Thank you for coming and joining us for this symposium on diffusion MRI. I will talk to you about connectomics and tractography and parcellation, like integrating some of the techniques we learned this morning. And if you have any, interrupt me during the talk or send me a question to Twitter or by email and try to respond even online. I don't know if this works. Okay, so what is the connectome? Well, you heard about it, and it was mainly introduced also by Sebastian Seung. So let's go back a step in the 90s, where we had like the genome. So everyone was working on the genome, and now we're working on the connectome. So in that time, well, we thought like the genome would explain everything, like the personality, all your characteristics. But, well, we learned the genome is not enough. So are we more than our genes? And, well, Sebastian Seung proposed this, and, well, he says that I'm more than my genes, I'm my connectome. So what is the connectome? Well, the connectome is a network first, like the connection between the neurons in the brain, and it describes like the structural connectivity in the brain. And all this connectivity can be done in two different levels, like first on the structural level and on the functional level. And on both levels, we have to differentiate between different scales. So one of the scales is like what was introduced here by Jeff Lichtman. So the microscopical scale, where you have like the neurons and the axons and this microstructural connectome, like the connections, really the synapses between the different cells. But what we are looking on is the axonal projection, so a different scale of the connectome. And the functional connectome is based on the dynamics of the pattern of activity, and it just analyzes statistical dependence of the coherence of the activity, and then you get like networks like this what was proposed by Dosenbach and others as the functional connectome. So we focus on the structural connectome since we can do it with diffusion. But first of all, what we need for the connectome, first we need nodes and edges, so the endpoints and the connections, we have to characterize them. So on the micro scale, as done by Moritz Helmstetter from the MPI in Frankfurt, he defines like on the micro scale the nodes as the neurons and the edges as the synapses. So these are the connections. If you go to the mesoscale, then the nodes are the columns, and if you go to the macro scales, then the nodes are cortical regions, and the edges are the white matter pathways. Well, and so we have to keep in mind that connectomes exist at different levels, and that if we do it in diffusion, we also should look what our biology neighbors do in this microscopical scale and how to get this together. Just very few people do this interaction, so maybe we can step in this direction also in future. So what are the basic components? Again, well, we need the nodes, which are on the macroscopic scale, is the parcellation. One of the parcellations was proposed by Brodmann, like in the 19th century, like based on site-like tectonics. A different parcellation was based on the gyrification by the free surface software and others, which just identify the different gyri and then assume that each region which has a specific color has a specific function and has specific connections. This is something I would like to question a little and maybe use a different method in this to build up the connectome. So then the next step is the edges between, so this is the white matter, so the connection between the areas. Well, this is we learned about, and then we get together to get this connectome. And, well, thanks to this nice picture from the Connectomics group here in Lausanne, close by, and which get together these networks. And what I would propose is to do both the edges and the nodes based on diffusion MRI. Therefore, we do like diffusion MRI based parcellation. So how can we do this parcellation? The parcellation is based on similarity of tractograms and differences in tractograms. We know from the monkey literature that different areas in the brain have different connectivities. Like here in this picture for two different functional areas, and the connections to, well, at the border of the spider plots we have different functional areas, and this is just the spider plot, the connection strength for one area to other areas. And we see that for these two areas on the left and the right, they are different. So, and we know that within each of these areas, they are homogeneous. So we can exploit this and use the same idea in diffusion tractography. So define an area by its connections. So we saw this way of tractography. So here we propose, like going from left to right, to put seed points at the white matter gray matter boundary. These are the green dots on the left, and then we do a tractogram, like here in this case a probabilistic tractogram to the whole brain, which is shown in the middle column. And for the different seed points, we get different connectivity patterns. And if we look on them just in a slice, well, we see this probabilistic connectivity pattern as we saw it in the probabilistic tractogram before. And so now, if we want to compute similarities between these tractograms, we can do it directly in the image space. So this means we had like two different tractograms, like here on the left and the right-hand side, and we just overlay them and compute the distance between these two tractograms. This distance is computed mathematically as just the cosine of the angle between the two high-dimensional vectors. Okay, well, this is very mathematical. In principle, it just shows us how similar are they, how strongly they overlap. Like here, the blue voxels are those who overlap between both measures. If they completely overlap, then the overlap is 1, so like the distance is 0, so they are completely similar. And well, we have a measure between 0 and 1. So if the two vectors overlap, if the two spaces overlap, then they are completely the same. And if we have now two seed points which are close together in the brain, we expect them to have similar connectivity, so very high similarity, and lower if we go far away. This similarity, I just want to point out, is very close to a similarity which could be done on the correlation between these tractograms. If you use the correlation as a measure, then you would have values between -1 and 1, which is not so nice with respect to the mathematical properties of the matrix you get out of it. So I talked about a matrix. What you see here is like this matrix which we get. So we have again like the seed points and the cortex. For each point we get a tractogram. From the tractograms we get the similarity matrix, which is already here in the middle reordered, and which groups together like points which are very similar to, which have a very similar connectivity profile in regions which are close together. So this can be done via, as proposed by the Oxford group, by Heidi Onsenberg already 10 years ago, by a spectral reordering, and then by eyeballing of the boundaries, or we can do this with other clustering algorithms directly on the similarity matrix. So the parcellation of the cortex is not based on the clustering of the tractograms, but on the clustering of the similarity matrix. So here we get, like for this area, like including motor, premotor, and pre-SMA area, we get 3 regions, subdividing clearly the motor from the premotor areas here in the mediofrontal cortex. This was just to prove it, but one of our focus in our institute is language areas, so we wanted to see if we find the tectonical boundaries which we have in the infrafrontal gyrus, also in this diffusion tractography. So we took a region in the infrafrontal gyrus and did the same procedure with a k-means clustering in 3 areas, and we got, like here, the green area the blue and the red area. The red area, which corresponds to like a frontal operculum, which has like mainly a ventral connection, as you see here in this connectum patterns. Then we have like the middle area, which is close to the pars triangularis, so which is Brodmann's area 45 for the experts in this field, which has a ventral connection and a little bit of a dorsal connection. So like you see here, like on the very right, you see these different patterns of connectivity which characterizes each of these areas, and which gives you the basis for the parcellation. So we don't rely on the absolute value of the connectivity, but just on the differences between the different connections. So we can do this for all brains, get relatively robust results, but we can also do it for other areas. And an area like here, which we saw before, is the precentral gyrus. And if you would ask Free Surfer, he would give you just one label. If you would ask the Brodmann's area or like the cytoarchitectonic maps, you would also get one or two area, like Brodmann area 4, which is the primary motor cortex, and partly also 6, so which is the premotor cortex. So this would be like functionally homogeneous, but we know that, for example, the blue area in the upper part is the motor area responsible for the foot movements. The green area is more the hand area, and the yellow and the red ones are face and mouth areas. So we know functionally they are different, and we wanted to test like if this functional differentiation can be also done by differences in parcellations. And what we did here is like we got this, for this precentral gyrus, the parcellation out, and we got these 4 regions out with different connectivity profile. If you see the tractograms on the upper row, the very right, you see like just mainly the corticospinal tract going down from the blue area, and well, from the red area you get like again this part of this motor speech network. So we have quite different patterns of connectivity, so we can subdivide this cytoarchitectonically homogeneous regions much more finer details. And if we go and we map this together with the functional activations, we had like 3 different tasks. One which is more like for the rhythm task, which is the left column. We see that the activation mainly falls in the ventral area in each individual subject. And then we have like an object area where it's more like grasping, so the hand area would be activated. And then we have a location task where the people imagine to walk to this place, so the foot area is activated. So this nicely corresponds on the single subject level to the functional activations which we get also in this area. So this means that this subdivision, which is much finer than the single gyrus, makes sense and should be used for connectomics analysis. So what do we want to do if we do bigger regions, not only one gyrus? Then we cannot do it with the simple k-means approach. So what we proposed here is to build this in a hierarchical way. So just as an example for a small region again, so we do seeding at the gray matter white matter boundary. We do the similarity of the connectomes, of the tractograms, but then we translate this in this hierarchical tree where we put the similarity between 2 points in, we just like rearrange this matrix where 2 points which are close together join very early, and they build up this tree. So the tree stores the whole information which is stored in the matrix. And then from this tree, we can cut it at a certain level and go back to the parcellation. So the parcellation you see here on the inferior parietal cortex would be similar if we should just do a k-means clustering of the 3 areas. So how does this work? Like first we start on the single leaf points on small areas which cannot be separated anymore, and then we look for those leaves in the neighborhood which are closest together, and we join them. So 2 voxels are joined together, and they form on the first level a new tractogram. We compare this new tractogram to all our leaves, so all single nodes, and in an iterative way, we build up in this way the whole tractogram. So the whole tree of similarities. So they are joined together, they build up bigger regions. We compare them again to the neighbors. This is what is called agglomerative hierarchical clustering, and we build up the whole tree in this way. If we have such a tree, and we can take like a region, like here the green region on the upper left, out of the tree, and then we can navigate through this tree and cut it at different levels. So if we go to the upper right, we cut it just in 2 areas, and then we lower this threshold, we can cut it in 3 areas, or like if we lower the threshold farther, like we can subdivide this infrafrontal area in smaller regions. So we can interactively navigate through this and explore the hierarchical tree structure. If we go through this structure on several levels, just for the whole brain, here is an example. If we use a very, very coarse structure, as here for 15 regions, which is on the left, we see that we find like big areas, like the red area on the lower end, which goes along the temporal lobe. They all share a very strong network of connection of the inferior frontal occipital connections and the inferior longitudinal connections, and then we see this light blue area which corresponds to this area which are close to this arcuate fascicle, the language network, which also clustered together. But this is not really meaningful, but if we go to 50 or 100 region, we see already a parcellation which gives us areas which we know are functionally relevant and which could be used for further analysis of the connectivity pattern and the matching to functional areas. I just want to point to a further work in this direction by our colleagues from INRIA in France, from Damien Wasserman and his PhD student Galeo Diaz, which has a talk on Tuesday and 10:43 and a poster on Monday. So pass by this work. He just used this method to go a little step further. Well, if we have like this parcellation, then we can use every small area and get the connectivity pattern. And here we see like the connectivity pattern of like some of these frontal areas, and we see like that frontal areas mainly have local connections. So we can from each connection again get the pattern of connectivity and analyze this further. And well, of course, like we have implemented this in a software here as a module in this open walnut, so everyone can just put in his diffusion data in this, use like the standard tractography methods, and then explore this hierarchical tree. So this might be helpful for many people, and it's available on GitHub. But one little disclaimer is like, how sharp are this boundary between these areas? This is something which was discussed in the anatomical community since a long time. How sharp are these boundaries? In anatomical sense, many people claim for sharp boundaries. Can we see sharp boundaries in diffusion? And thanks to the work of Leonardo Cerillani, and he pointed nicely out that this boundary can be sharp, as here for the SMA, pre-SMA separation. So here he mapped what he called like the Laplacian eigenmap. So it's a sort of a gradient of similarity. So just by reordering this matrix and mapping it back and getting like the similarity between the tractograms and getting like the first mode of this similarity as a color map, you can see like how sharp the boundaries, the similarities change, and how sharp a boundary is. So for this pre-SMA, SMA found quite sharp boundaries, but for other areas like here the insula, which was one of his areas he worked a lot, we don't find sharp boundaries. So we don't find, well, we still have clear differentiation in the connectivity profile between the anterior and the posterior insula and between the dorsal and the ventral insula, but you cannot say that there is a sharp boundary, like this has this function and this has this function, and there is a sharp differentiation. And this is somehow important, as you can still do a subdivision of this area, a quantification, a characterization of the connectivity profile, but there is no clear evidence for parcellation at this level for the insula. There's something to follow up, and there's something to keep in mind, also like for the connectome analysis, that the boundaries are not as sharp as we would like to have them sometimes. Okay, so, well, now we have the parcellation more or less sharp. How do we go to connectivity? Well, how can we quantify this? And if you can quantify the connectivity and use this, then you might go to nature neuroscience, as it was done in many studies, like here, where they correlated the connection strength with behavior, just as personal behaviors like novelty seeking or revert dependency, you see, like nicely correlates with the connectivity strength between the hippocampus and the amygdala, with the caudate nucleus. If you find something like this, then this is great, of course, like people will like it, but we have to take care, as we saw before already in the tractography methods and in the connectivity analysis. I show you some of the weak points. You have to take care, even if you have such results, think twice before putting them out. So what are the weak points? And, well, first, how do we quantify connectivity? Well, many of you know, like, we just take regions, do tractography, and then we compute, or we can compute, like, the relative number of streamlines connecting one area with the other areas. So relative means, like, we have to normalize by the number of streamlines we see in area A arriving in B, and by the size of area A and area B. This would, like, in biological terms, mean, like, what is the amount of influence activity in region A would have on activity in region B. So this is something we can quantify, but we have to take care. So one of the points, many of the points were already focused on, but I want to go a little deeper. So, like, if we have here in this example of the inferior parietal lobe, we saw, like, the connection to the frontal lobe. So the parietal lobe is here on the right side. We look at the connections to the frontal lobe, and we have, like, two target regions in the front here. Well, we can now count the streamlines arriving in this area. But we have also streamlines which are like this, and they cross both areas. So what do we do there? How do we count them? This is a very, very big problem, since many of the pathways just cross underneath the white matter gray matter boundary, and so these areas, we get it from the atlas, might be crossed by many of those. Since the streamlines don't stop when we enter one area, we can count them twice. Should we count them twice? This is also particularly important for subcortical areas, but also for cortex. So this is one point. A second point we see here, like, where do the streamlines end? Well, we have most of the endpoints at the crown of the gyri, like you see here at the very tip of the gyrus. And this is something which is called, like, the gyral bias. There was a publication on this, and I go to it a little deeper. And the third point is, which wasn't discussed so deeply, is the distance bias of connectivity measures. So, like, if you see it in a certain area, you have a lot of connections very close to, and the farther you are away, the weaker the connectivity is. So this influences your connectivity measures dramatically. First, going to this gyral bias. Well, you might heard of it. This was, like, nicely illustrated in the paper by Rivley in PNAS last year. So if you have, like, a high resolution scan here of the gyral crown, you see that the diffusion properties are more radial. And if you go to the fundus, Then you see more tangential diffusion orientations, which might be related to U fibers connecting the two gyri, or by other effects, which just prevents the streamlines from entering in the fundus, so deep down, like here in these areas, they just pass by. And what is the result? Well, as illustrated here in this monkey brain, most of the streamlines just end at the crown. And if you want to differentiate connectivity pattern, and you just have the unfortunate position that in your task you must differentiate between the fundus and the sulcus, and the crown and the fundus of a structure, you will get differences. This is something which is not really solved and not with standard tractography algorithms. So take care not to interpret this difference. Another thing is like if you want to define your regions of interest based on the cortical parcellation, that the cortical parcellation must match nicely between the anatomy and the diffusion scan. This was already discussed also. Like if you do a registration between the diffusion images and anatomical images, as it's done here in the upper left, the red boundary is the diffusion image, it's distorted. So because of the EPI inhomogeneities of the B0 magnetic fields, they get distorted more or less strongly, and there are several different methods to correct for this. One is the field map, it is done from the fMRI, and the other methods is to acquire images with opposed phase encoding directions, as we heard before, and like we compared different algorithms to correct for this. Like these different phase encodings can be done with the implementation in FSL, which is the top-up algorithm, or like the implementation in the SPM toolbox from Siabosch and Las Rutotto in this HISCO method. They all show more or less the same result. They improve very nicely the registration to the anatomy, but still it's not perfect. So you must be aware that the connectivity pattern might differ between the posterior and the anterior wall of a gyrus because of the seed points are not perfectly matched, as long as you don't define the seeds directly in the diffusion space. Okay, so if we did all this, we did define it perfectly, the diffusion seed areas and the target areas. We compute the tractogram, like here in this for the inferior parietal lobe. We get certain target areas and we quantify the connectivity. And then you can see, like here in this example, that the connectivity pattern between the anterior, medial, and posterior area in the parietal lobe strongly differentiate. So this is the red, yellow, and green with respect to the connectivity strength to certain areas, and they also differ between left and right hemisphere. And we see like that the connectivity strengths are different between connections to the frontal and the parietal areas, which are just in this plot. You don't need to see to read the details, but just see that there are differences. But if we zoom in a little, we see that these differences, like here, for example, on the left, for the connectivity to the suprafrontal gyrus, they vary between like 1 and 1,000. So between brains, the connectivity strength varies with a factor of, several magnitudes. And if you want to correlate this with behavioral measures, you will have bigger problems, since, well, I don't think that some people are 1,000 times more novelty seeking than others. So there are differences, but not 1,000 times. So correlation with behavior is difficult because of this high variability of the connectivity measures. So be careful if you correlate this with behavior. And even if you do this with the best method available, like we did here, 7 Tesla diffusion MRI with 1 millimeter isotropic resolution, prop tractography from several seed areas in the frontal lobe to the thalamus here, well, the first good news is that they could find this connection in a relatively strong way in all brains. And if we plot them in the logarithmic way as here in this figure, then they all look nice. But if you plot them with linear scale and you look on the arrow bar, for example, in the left, then you see also that the variability across subject scales with a factor of 10. So in this study, we didn't look for a correlation with behavior, just if we can reconstruct the connection, and therefore it doesn't matter if we have like 10,000 or just 1,000 streamlines connecting both regions. But if you want to quantify across subjects this variability, you must be aware of some sort of variability in the tractography itself. Okay, the other thing is different local models. We saw this, although different tractography methods like probabilistic and deterministic also influence the results. And there we just systematically varied like different local models like tensor, multi-tensor, ball and stick from FSL, spherical deconvolution, and persistent angular structure with probabilistic and deterministic tractography, and did connectome analyses with the different methods. So we defined like here a set of about 10 seed regions in the left and right hemisphere, which are like in this language network. And we looked on the connectivity matrices computed with the different method, and also represented the data as a connectogram, like the connection strength in such a network representation, where you see like the left and right hemisphere and the mid-sagittal areas, and the connection strength between them, color-coded, like red is very, very strong connection, blue is medium, green is weak, and absent is not shown. And if we compare this between the different methods, you see this very, very well. Of course, there are connections in all brains, so this is good news. So most of the algorithms find similar connections, but if you look on the difference between these connectograms, you can immediately see that there's a big variance between, for example, like in the upper row in the middle, this deterministic persistent angular structure tractography, which nearly finds connections between every node, and like on the left top, the tensor deterministic tracking, which finds relatively weak connections. And there is something like in between, like FSL methods, which are relatively robust and seems not to be prone to too many tracking artifacts. So this comes back to the points Maxime Descoteaux pointed out earlier, that there are a lot of false positive connections and false negative connections. So depending, and this also applies to the connectome analysis. So it strongly depends on the method you use. And can we do different quantifications? So it was already mentioned that we could do like along the tract analysis. This will be done later in today again with like T1 relaxation times or fiber density measures. And I want just to propose like some things which was really recently published, like the work from Alice Lebois in her PhD thesis from Neurospin. There they measured quantitative T1 and T2 relaxation times and mapped them on the fiber bundles, once like along the tract, as you see here in this along the tract values, and also like for the whole tract to put it then in the connectome analysis. And this might give us more relevant information than just the fiber count. So getting this measures which really represent the diffusion, the microstructural properties. Also, well, of course, you can do something like FA, but we know FA has problems in crossing areas, and to map this. To overcome the problem of crossing areas and FA, we've proposed new methods based on diffusion measurements, like here, based on spherical deconvolution, and we did something like FA for each of the lobes, for each of the directions, by approximating the lobes of this fiber orientation distribution function with single fiber orientation distributions, and then get quantitative measures of the different bundle properties and to replace FA. So if we do this, we have like here for 4 different voxels, one with a crossing area and one with a single fiber. We can see that the fiber fraction, which is like how many fibers are in the first and the second directions, and varies between the different local models. And we can see that with these measures we can quantify the number of fibers in each component and map this back in the volume and map it back also on a track. And we see here, for example, that the fractional anisotropy in these crossing areas, they just drop down dramatically. But this is caused by like the fiber fraction, so a lot of fibers in the secondary directions, and also a strong increase in the spreading of the fibers in this area. So if we have this, then we can go to the network, and this is somehow like this kind of network analysis is in a different session. Therefore, I don't want to go too deeply in this and just introduce and acknowledge the work of Patrick Hagmann here from Lausanne, who was pioneering in this network analysis and proposing like a fine subdivision of the cortex in small areas, and compute like the connectome from this. And they also compared like different levels of connectivity, and showing that the connectome matrices strongly depends on the size of the regions. One of the points is, for example, here if you have a big region like the green and the pink region, they just have a small subpart of the areas are connected by this arcuate fascicle, as you see in the smaller areas down there. And if you quantify with the smaller areas, you get different connectivity profiles if you use the bigger ones. And once we have this, well, we can go then the next step, as proposed by Oliver Spons, to do this network analysis, subdivide the network in modules, get for each of the modules some parameters like the path length and the clustering, and do like local and global measures, like the local measures of integration, how important is a certain node in a certain network, and how strong is the node embedded in the community of networks, and also global measures for the whole network, how easy it is to reach from one point to another point, and how subdivided is such a network. And using this, well, we know that the brain is not homogeneous, so we have regions which have a higher degree, so they have higher connectivities. These are the red big dots, and they are organized in a way which is close to this default node network. So we have this parieto-frontal connectivity, or areas which seem to have a stronger connectivity also in this network characteristics. With this, well, I want just to summarize that diffusion MRI allows to study the brain connectivity and allows to parcellate the brain. With network analysis, we can capture the complexity of the brain, and the integration with cognition is still very, very challenging. And then I want to thank you for your attention and my collaborators. And thank you.
Good. Thank you, Alfred. We have still time for 2 questions. Yeah.
I think it's very interesting, and I don't mean to sound critical or anything, so I just want to put it out there. I just have a question. Has anybody tried to do reproducibility studies on connectomics or connectomics? What I mean is, when you try to track something long range, then you're really sensitive to propagating errors. So even if in every voxel you have 0.5% error, by the time you get to the 1,000th step, you can be really far off from your true target. And so one thing that would be possible to do to modulate the errors, so for example, eddy currents or susceptibility-induced distortions would be modulated if you put the subject in, let's say, on their side. Like once you scan them lying on their back, once you scan them lying on their side, and can you reproduce the same results? It's just a question. Has anybody done something like this?
Well, like testing, retesting was done, but normally like they put them in the same way in the scanner, and actually like if you rotate the brain in the scanner, the distortions change completely, and also the way you can correct for them. And so I agree that this is a big issue, but this is not the only weak point. Like in the same brain, you are more highly to get the same tractography still. Since something like the gyro configuration seems to influence this tractography tremendously, like if a gyrus is deeper or not, so you get different connectivity values, and this is something which cannot be addressed with this. So, yeah. But there are studies which try to do a retest measurement, but not to the extent you would like to have it. Yes.
Right, yeah, no, I was just wondering. I mean, I agree with you that the distortions will terribly change if you turn left as opposed to face up. But I don't see why the correction methods should change. You would apply the same correction methods, right? Like, let's say blip up, blip down for susceptibility distortions, and the eddy current, the eddy tool should.
Yeah, but these correction methods are never perfect. So, and if you're in an area of strong distortion, they cannot do, like if you have a signal drop out, they cannot reconstruct it. So, and if you like just rotate it by 20 degrees the head without changing the field of view, then the distortion will change dramatically, like in the frontal lobe. And this also.
Okay, thank you.
Thank you.
More of a practical question. So if in our study we were to do the parcellations based on the connectivity, how would you advise we go about it? Would we first create like a group map, combine the data from all of our participants and do the parcellations on a group level and then map those regions back into native space of subjects? Or we would just create a parcellation in the native space of each person?
So if you are interested in the variability between the individuals, then of course you cannot map them in the group. If you want just to have like an overall subdivision for like a template, as like for the free surfer template, get all the gyri, and in this case like a robust, then you should do it of course in the group template. So like do the tractography in the single subject space, and then map them in the group template to do the parcellation, and then project back in the free surfer way to the single subject. If you want to compare the similar regions and then do a connectome analysis on this. But there is still work to be done to do this robustly across subjects. So
no ready pipeline for that.
Yeah, I'm sorry, no. Since it's questionable how meaningful is also this variability. So I think there is quite some variability in the size, in particular, like of higher cognitive areas, and like using the same parcellation is maybe the wrong approach. In particular, with respect to the strong variability of the gyrification and the interaction between gyrification and connectivity. So therefore, it's not a simple problem. Thank you.